students, welcome you back to my channel BIOMICS Hub. Hope you are doing well. So in this today's video we are going to discuss about the signaling pathway of another 152 hormone which is known as strigolactone. So what is the role of strigolactone? The role of strigolactone is that it helps in the germination of the seed of a parasitic plant which is known as striga. Except the germination, what it does, it also helps in inhibiting the branching and it helps in promoting the rhizosphere interaction. Okay, it helps in the symbiotic relation between the arbuscular mycorrhizal fungi as well as some nitrogen fixing bacteria. Due to the symbiotic relationship, what happens? There is a adaptation towards drought, and also there is that the nutritional stress is controlled, or there is nutritional stress which has been controlled by the plant. So we have to understand where is the location of the synthesis of the strigolactone. As we have already discussed in the last videos, that where is the synthesis of the auxin. The synthesis of the auxin was in the tip portion. So the transfer of this auxin was towards the base. Okay, in little amount the synthesis was towards the root, but the main part where the auxin is synthesized is the tip portion. Okay, but the strigolactone as well as cytokine, where they are synthesized in the root part and they are being transferred towards the upper region. Okay, so here we can see that the cytokine and strigolactone, they are being synthesized in the base region and it is being transferred to the upper region. Okay, now we will understand the main topic of our today's videos, that is how the signaling is taking place. So for the signaling, here the signaling is also by the E3 ubiquitin ligase mechanism. So the receptor here for the strigolactone, it is a hydrolase. It is known as D14 alpha beta fold hydrolase. Okay, what this hydrolase does, it reacts with strigolactone. It works as a receptor for strigolactone and now it is being activated. When it is binding with the strigolactone, it is being activated. After its activation, after it gets hydrolyzed and after the hydrolysis it gets activated, where it comes and binds with MAX2A, which is a F box, in conjugation with PSKS, which is a SKP, and Qlin. Okay, now it recruits the target protein. And it is being degraded. The protein which was inhibiting, it is being degraded. How it is being degraded? It is degraded by E3 ubiquitin ligase mechanism or by 26S proteasomal degradation by the use of this polyubiquitin. When this polyubiquitin is being added in the target region, it is being degraded. After this degradation, the strigolactone is being released from this D14 alpha beta fold hydroxylase, and after this D14 alpha beta 4 hydroxylase is released, now it is ready for binding with another strigolactone for the same process to be continued. This is how the signaling of the strigolactone is taking place. Okay, here the receptor is it is a hydrolase. Okay, so how the strigolactone, auxin and cytokinin is being regulated? We have understood that this cytokine which is present, it is being synthesized by IPT. Okay, this strigolactone which is present, what it does, it inhibits this IPT. So indirectly what it does, it inhibits the synthesis of cytokinin. Why cytokinin is inhibited? Auxin which was present, it was synthesizing the MAX4 and after the MAX4 has been synthesized, it was stimulating the formation of strigolactone. Okay, and this strigolactone was helping in the activation of BRC1 gene, which was inhibiting the auxiliary bud growth. Okay, so now the cytokinin which is present, it helps in inhibiting the BRC1 enzyme as well as auxin. Okay, so this auxin was also responsible for inhibiting the branching or shoot formation. It was helping in rooting. So by inhibiting this auxin, the cytokinin was helping for the shoot formation. Okay, this end product, the strigolactone, which was producing the BRC1 gene, which was responsible for inhibiting the auxiliary bud formation, the cytokinin will help in inhibiting this BRC1 gene. Okay, this cytokinin will inhibit the end product of this strigolactone, and strigolactone will inhibit the synthesis first portion, which is going to synthesize the cytokinin, that is the IPT. In this way, these three phytohormones are being regulated. Okay, I hope you have already understood this pathways, signaling pathways, where it is being synthesized and what is the correlation between these three hormonal signaling pathways. Okay, from this topic there are various type of questions which are being asked in CSIR Life Science Nets. In the next video we will also discuss about the questions which were asked in the exams. So thank you everyone for watching this video till the end, and if you like this video, please do not forget to like, share and subscribe. Thank you.